So a warm welcome to you all participants to today's e-seminar on genetic testing in kidney diseases, which is organized by the Working Group of Inherited Kidney Diseases of the ERA EDTA, and which will be held by Professor John Servais, which I will introduce to you in a few minutes. So today also present are the panelists, also from our working group. These are Drs. Aude Servais and Emmanuelle Plaisier. And my name is Knoers. I will be your moderator today. So before I introduce Professor Servais, I have a few announcements to make. First of all, by participating live, you are able to earn 1 European credit for your continuous medical education. And this is an exclusive benefit for ERA EDTA members. And the next announcement I'd like to make is that the next working group e-seminar entitled Volume Management in Dialysis, Current Perspectives in Clinical Practice is scheduled on April 15 and is organized by the European Dialysis Working Group. So today's e-seminar will be held by Professor John Servais. Professor John Servais is a professor of renal medicine in the Institute of Genetics Medicine in Newcastle's University. And he is a honorary consultant nephrologist in the Newcastle upon Tyne in NHS Foundation Trust hospitals. John's research is focused on the genetics and molecular mechanisms underlying inherited kidney diseases, and particularly cystic kidney diseases and tubular transport disorders. John has discovered numerous novel genetic causes for these disorders. And his work has been published in more than 170 articles that are published in peer-reviewed biomedical journals, among them many with high impact. John is also a frequently asked speaker on international scientific meetings because of his knowledge on genetics and nephrology. Today, he will present his e-seminar, Genetic Testing in Kidney Disease. We are very much looking forward to your e-seminar, John, and the floor is yours now.
Thank you very much, Nina. Thank you to everyone that's joined this session, and welcome to those that are watching it back on video, having access to it. So I'll attempt to share my screen just now. And hopefully that's projecting okay. So as Nina kindly said, I'm a nephrologist in Newcastle upon Tyne in the northeast of England, and very much still feel part of the European Union, even though the politics of that is become messy. But I think the arena of renal research, in particular genetic kidney disease research, relies on collaborations across the world, and so I very much see myself as part of Europe in ongoing collaborations. So what struck me about this topic when I was preparing for it was the sort of huge range of things I could be talking about. Am I talking about genetic testing in Alport syndrome, for example, or in cystinuria, or in glomerulonephritis? And so I've had to restrict, for reasons of time, some of the diseases I talk about, but we can open up the discussions, and I'm going to make sure we leave time for discussion so that if I've not covered a particular topic, then you can ask about it. So just struggling to find the next slide button. Here we go. So by way of background, I want to talk to you about genomics. And what is genomics? Well, it's kind of a made-up word, isn't it, from the term genome, and we've applied X to transcriptomics, genomics, metabolomics. And so genomics is a sort of catch-all phrase for everything from a single point mutation in a single nucleotide base through to a whole chromosome missing and everything in between. So we consider at this stage the point mutation stage, a micro change in the 3 billion or so bases we have, up to bigger changes. But any of these changes could cause disease and are part of the term genomics. And so if we consider this spectrum, then down, and just see if I'm going to get a pointer here. Down here, at this end of the spectrum, we're dealing with rare and ultra-rare Mendelian diseases, whereas, you know, other variants within the genome may be more common and may give you a risk allele to a certain disease. And moving up towards the other spectrum, there may be just polymorphisms that don't actually do anything, or that we know about are part of normal variation. And so this defining normal has been taking place over the last 300 years. So what do I mean by normal? Well, if we define normal, it's very important, because if we're looking for disease variants, then they are abnormal compared to the normal. So we need to know what exactly normal is before we can describe patients as being abnormal. So defining normal took 300 years. Cracking the genetic code, as I'll show you, took considerably less, 3 years. And now this era of genome sequencing happening very rapidly, maybe over the last 3 years or so. So time is moving very quickly. So here are the 300 years that I'm describing for us to define normal. So we've got the first microscopes being used by Hooke and various other people to look at cells for the first time and describe cells and nuclei from cells. And then we have Mendel here, 1856, describing genetic information being transferred using his P's, etc. And here in 1889, we have the first mention of traits that are being passed down. The word gene wasn't introduced at this stage, but that's kind of what the implication was. And, you know, moving swiftly forward, we've got Watson and Crick, 1953, describing the structure of DNA and the double helix that has become very famous. And so, you know, it wasn't really until 1956 that really we defined genetic information in 46 chromosomes in humans. So, you know, this history to me is fascinating. Fast forward into sort of the modern era from the 1950s through to the present day, then lots of things are happening. And so we have largely the invention of techniques to look at DNA. So we've got southern blotting, and we've got Sanger sequencing, which really was a game changer in terms of us about being able to look at DNA. We've got the description of micro deletions. Get rid of that. We've got the description of micro deletions, and, you know, a big sort of landmark was the first chromosome to be sort of fully mapped, and the cystic fibrosis gene identified on chromosome 7. And so alongside these sort of technique discoveries was the implementation of genetic services. And I, as I've said, work in the UK, and so we have the NHS, which then, you know, lags behind research discoveries, but actually uses these genetic techniques in terms of genetic diagnostics. And so, you know, we have this hugely important 13 years of the Human Genome Project here that started in 1990, finished in 2003, and cost £3 million to sequence one whole genome. Interestingly, this started from, you know, an advert in the town of Buffalo in New York State that advertised for volunteers to have their genome sequenced, and 10 people came forward, 10 males and 10 females, 20 people, but they ended up using only a handful of those people to actually come up with the first blueprint of the human genome as we know it. And of course, you know, there's been various iterations of the human genome since then, using more and more different individuals to contribute. Okay, so we can approach study of genetics by two angles, really. So there is this phenotype-driven diagnostics, in that you have a patient in front of you and you carefully look at their phenotype and then decide what might be the likely genetic causes of that particular phenotype. And this approach will minimize the amount of variance of uncertain significance and incidental findings that you might come across if you use a wider approach. And it has a greater sensitivity. So that's one approach, defining a phenotype. The other approach that we've seen has become more and more popular in recent times is this genotype-driven diagnostics. You know, just sequence everything, do an exome, do a genome. In that approach, we create a large amount of data, including variants of uncertain significance and incidental findings. The sensitivity of the test might be go down. There's issues around consent, issues around information technology and bioinformatics. And so, but the more and more we do this genomics investigation allows us to map what we're seeing at the genome level to what we're seeing at the phenome level. And so I think, you know, both approaches are valuable, but they have to be linked up. The approach that the NHS has taken, largely to save money, I guess, is this utility-driven diagnostics. So basically, there's a cost-benefit analysis taken in terms of the diagnostic pathway. And so different techniques are applied to different situations. And so you might do a gene panel before you do full exome or genome sequencing, or if you're considering many different genetic causes of a particular syndrome, you might just approach this from a genome or exome point of view. The point I'm trying to make is that whole genome sequencing, although very powerful and very useful, at the moment doesn't seem to be always the most appropriate starting point, and there may be smarter ways that we can get to a genetic diagnosis. So this is what the NHS has done. It's moving away from single gene sequencing towards panels and virtual panels, applying the technologies of this rapid next generation sequencing to exomes and genomes. But there will be always a place for bespoke Sanger sequencing, which is the gold standard to assess variants. That will still be required to verify stuff. The old-fashioned sort of counting chromosomes and FISH techniques are moving towards array CGHs and SNP arrays, looking for deletions in chromosomes. And in the NHS, there's been a centralization of services to allow greater efficiencies. Okay, so going back to this thing called the genome, I mentioned there's 3 billion nucleotides, but we all vary from one another by about 40 million different alleles, different base changes, nucleotide changes. And so you or I could be compared to the reference sequence and find 38 million different changes. And so this is what I mean by defining normal, in that we need to know more information about different populations, different ethnic groups, different people, about what is an acceptable variant from normal and what is disease causing, what is not disease causing. In each individual, we might carry 1.5 million small indels. We might carry many thousands large deletions, but our phenotype remains normal. And we can go down to a more sort of loss of function level, and we all carry around 150 loss of function variants, 2 to 5 of which predicted to be damaging, and yet phenotypically many of us don't have these diseases. So the genome isn't the be-all and end-all of it. The body has found ways to adapt to loss of function variants by alternate splicing and different levels of expression and redundancy in genes that we're still trying to figure out. So I think as an introduction, really, I'm just laying on thick the complexity of the genome and what we're up against. So moving on to the title of my talk, really, genetic testing in inherited kidney diseases. As I alluded to, you can talk about each of these specific subsets of diseases. And it is true that, you know, different parts of the world have developed different sort of specialty interests in these conditions. So here in the UK, Bristol would be known for its work on glomerular diseases, whereas other centres would be known. Newcastle's particularly known for its work in hemolytic uremic syndrome. And so, there is this concept of developing local expertise in order to understand these genetics of these rare inherited diseases. Okay, as I alluded to, the approaches can be targeted or wide. So panel sequencing might include a couple of hundred genes. Whole exome sequence includes all the genes, 20-odd thousand genes. And then whole genome sequencing includes all the genes and all the intronic regions and non-coding regions. So panel sequencing is rapid and highly accurate. Exome sequencing is a bit slower and may not be as accurate. And then whole genome sequencing tended to be slow, although we're speeding up, but we've still got this challenge of understanding all the data. If you want to take home and read something from my talk, then I recommend this publication by Emily Grootman and colleagues from New York. And this really is a tour de force of explanation of how you apply genomics to kidney disease. And it takes you through all the steps of approaches with next generation sequencing and then understanding the results. I'm going to touch on a few of the themes from this paper, but really it would take me 2 hours to sort of dig into everything that this paper offers. But I encourage you to go and look at that if you want to have follow-up reading from this session. Just to remind you, inherited kidney diseases are common if you group them all together and account for 10% of adult end-stage kidney disease and 70% of all the nephropathies that our pediatric colleagues see. And aside from rare disease, the heritability of something as commonly measured as GFR is thought to be, around 30 to 60%. And so we all carry genetic variants that result in different levels of kidney function, essentially. And what's fascinating is that, up to 30% of adults with end-stage kidney disease report a positive family history of kidney disease. So by studying these diseases, we can learn lots about our patients and about the relatives of our patients. Okay. As I mentioned, Sanger sequence is the gold standard for confirming next generation sequencing finding. The chromosomal microarray is a very useful technique to detect deletions and changes in copy numbers, and it's probably the first place to go to for conditions such as intellectual disability, autism, and multiple congenital anomalies seen, for example, in children on antenatal scanning. And CACUT actually is a good example where copy number variants can explain around 10% of cases, including this fairly common chromosome 17q12 deletion that includes HNF1 beta as a good cause of CACUT. Panels can be very effective and fast, becoming first line rather than single gene things. And panels have been used for the investigation of this group of diseases called nephronophthisis related ciliopathies, as I'll come to later. But its panels alone can leave gaps, and you can miss variants such as deletions that need to be looked at using NLPA or chromosome microarrays to identify these changes. Okay. And then moving on to whole exome of whole genome sequence. Their advantages, they're completely unbiased. You aren't relying on a set of genes on your panel to look at a condition. You're saying, okay, let's sequence everything and see what we get. They also allow you to go back to the raw data and reanalyze them, reapply a virtual panel, if you like, or relook at certain mutations that were purported to be pathogenic previously, but are now thought to be non-pathogenic, etc., and vice versa. So I think whole exome and whole genome sequence do offer advantages, but the coverage of the genome is lower, and subsegments of the genome are difficult to sequence by this technique. WT1 would be an example, but actually we're moving towards doing an exome or doing a genome first line more and more, and it's quite interesting. And PKD1, this notoriously difficult gene to sequence using Sanger sequencing because of all the duplications, whole genome sequencing allows quite an effective way to sequence that gene. Some genes are really hard to detect whatever you do, MUC1 being a good example. Long-range PCR approaches are applied to this. Whole genome sequencing, costly, data storage issues, clinical interpretation and follow-up, as I said, but becoming the way forward. And if we consider this group of diseases, nephronophthisis related ciliopathies, there's 90 genes involved. And so, applying a panel and updating a panel is a lot of work. For example, this series of publications from the Hildebrandt group show that, looking at a panel solved 12% with a 13 gene panel, 21% with a 34 gene panel, 60 to 70% with whole exome sequencing. So you can see people are realizing that to detect these new genes that are contributing to these phenotypes requires a wider and wider approach. Okay, so I'm going to move to some case studies now, and we're going to use the Q&A chat facility, Q&A facility for you to interact if you want to. I'm going to leave some time for you to sort of interact with these cases. I've picked these cases because they happen to me, they're real life, I've not made anything up, and they're good examples of how you can get it wrong, completely wrong, even though you're trying really hard. So this is a family that presented to my pediatric colleagues. So there's a female proband presented at nearly 4 years of age with pyrexia and raised neck lymph nodes. And on sort of blood screening of this child, abnormal UNEs were noted. She had a reduced appetite and had not been quite right for some 2 years. The GP had noted some protein in the urine, but there was no infection. So the child looked pale, had some hypertension, had severe anemia. I think what this points to, well, tell me what this points to. You can put it on the Q&A. The urea is severely raised, the creatinine is severely raised, there's abnormal LFTs, and the GFR worked out by a Schwartz formula was 19. So this child at nearly 4 years of age has got severe kidney failure. So I think the low hemoglobin points to a chronicity of whatever's going on here. This is not AKI, this is a chronic kidney disease. So the abdominal ultrasound was done next, and both kidneys appeared small with increased echogenicity and showed no corticomedullary differentiation. My pediatric colleagues seemed to biopsy everyone actually, and so they biopsied this child, and the histopathologist showed an end-stage kidney disease pattern with sclerosis, glomerular fibrosis, so not immediately diagnostic biopsy. And so the underlying cause was uncertain, but my colleagues seemed to be fascinated by the levels of hypertension in this child and less fascinated by the ensuing end-stage kidney disease, which I'm not quite sure why. So the child was given antihypertensive agents, and the blood pressure was actually really, really difficult to control. This was quite unusual, I think, and they were challenged by this case. And ultimately, the child underwent bilateral nephrectomy, actually, which resulted in a resolution of the hypertension. Occasionally, we do this in adults, but it's become less common, I think, recently. And the child is now transplanted and doing really well. So that begs the question, what the original cause is of the kidney disease. What's really fascinating is, whilst this child was being sorted out, her younger sibling was admitted to the hospital. So day 3 of the girl's admission, the younger brother was admitted. He was only 13 months of age, and his creatinine was abnormal at that stage, being raised at 67. These are micromoles per liter, these units, giving him a Schwartz formula eGFR of 56. So we've got 2 children now with decreased kidney function in the same family. Obviously, we're thinking about which mode of inheritance. I guess recessive is clearly the mode of inheritance to think about here. And the second child had, again, a very similar echo pattern in the kidneys, increased echogenicity, but preserved corticomedullary junction, presumably because the child is younger with more preserved kidney function, and a degree of proteinuria, but not massive. And again, the second child, the boy was biopsied, it showed chronic non-specific tubular interstitial damage with ischemic changes in glomeruli consistent with hypertension. So rightly or wrongly, I think the pediatric nephrologists were thinking of an inherited cause of hypertension in this child. The outcomes of these children, as I mentioned, that the older girl got a transplant and she remains well with good transplant function. The younger brother followed a very identical clinical course to his sister and had severe hypertension leading to increase in kidney damage and resulting in both end-stage kidney failure within 4 years of life and a renal transplant subsequent to that. So a gene panel was done to investigate this family, and the panel was selected as to be severe early onset hypertension. And so this is a list of genes that were looked at, including mostly dominant conditions. Actually, you can see little syndromes in here. The autosomal recessive apparent mineralocorticoid excess gene was in there. These are Gordon syndrome's genes, but really, in my mind, I don't think this was the right panel. Well, I'm telling you, it wasn't the right panel, but because it was extremely narrow, very focused towards the blood pressure, and really didn't address the end-stage kidney disease in the two siblings, and didn't really address the recessive nature of the condition either. So this was negative, no surprise. This child was recruited into 100,000 Genomes project, and again recruited to this domain. So this project was running in the UK, and you had to say a phenotypic domain. So they were recruited this early onset hypertension domain, which maybe is not unreasonable, but that was reported as no pathogenic mutations. So long story short, it was only really when we unmasked the whole data set and said, okay, let's forget about this hypertension story and let's look for genes that cause kidney failure, that we found biallelic changes in TTC21B. You might not have heard of this gene, but it's a known cause of nephronophthisis type 12. It can also cause a proteinuric phenotype and can also cause a multisystem disorder called Jones syndrome, which gives you thoracic rib dysplasia. And so this family wasn't really solved until the whole genome was investigated rather than a targeted narrow approach. And showing here are the gold standard Sanger sequence chromatograms. Okay. So learning points from this case, I think accurate phenotypic data is vital. Use a renal biopsy and pathology help to guide you sometimes where available, but in these cases, I don't think it was helpful. Additional phenotypic data like urinary concentration ability and extrarenal manifestations may have helped. And consider the evolving clinical picture. So hypertension was big on the agenda initially, but kidney failure was also very big on the agenda, but happened subsequently. If the gene panel's negative, widen the search. Now, if time allows, I've just got one second case to discuss before we open up this discussion. So here's a family where there's a female affected again, presented at 12 years of age with fever, abdominal pain, but settled spontaneously. But interestingly, the admission bloods again showed a very low EGFR that was completely unexpected. Ultrasound scans showed bright kidneys and increased reflectivity. The urine was bland. There was no family history of renal disease, no other past medical history other than some autism. And the renal biopsy showed this, again, tubular interstitial damage, consistent with nephronophthisis. And this girl was recruited to the 100,000 Genomes project to allow whole genome sequencing. But again, no gene mutations were reported by the 100,000 Genomes project. When I looked at what panel this patient had been recruited to, there was a kind of misunderstanding of her phenotype. So she'd been recruited to this virtual panel domain called nephrolithiasis, which sounds like nephronophthisis if you say it quickly, I guess, but in essence, the wrong virtual panel had been applied to this girl. And so no NPHP genes had been looked at, and they'd issued a negative report based on the phenotypic data that had been inputted. Similar to the last case, while this is all going on, the older brother crash lands on dialysis. He's 18, he's feeling unwell while he's doing his exams in high school and gets a U&E done, and his creatinine's 2000. And so this prompted me to really look at this family again and manually look at the raw data myself. And this is what the BAM file looks like. So this is the raw reads across the genome. And here there's a massive gap in any of the reads all across here. And this is basically a homozygous NPHP1 deletion, which, as you know, causes nephronophthisis type 1 and would explain the phenotype. And so we went on to verify this. Is another fancy way of showing the deletion. So in color codes, in blue are the reads, in white are no reads. And so in this individual, there was a complete absence of reads across the whole gene. So I know Nini knows a lot about NPHP1 deletions, but we're able to show by MLPA and chromosomal microarray that the deletion was heterozygous in both parents, and sadly affected not only the two kids we knew about, but also the younger sibling who at the moment has got normal kidney function, but I suspect won't have for long. So the learning points from this case are very similar. Accurate phenotypic data is vital. Consider the evolving clinical picture in front of you in the family and other people getting phenotypes that perhaps weren't available initially. If the gene panel or targeted approach is negative, widen the search. Check you've corrected the correct gene panel, and then look for copy number changes, deletions. Consider array CGH and other approaches, looking for deletions if there's no obvious sequence variants detected. Okay, so I think I'll take advice about how many more minutes you want me to talk about here, Nina, but I'm happy to finish there and have a more general discussion.
I think there's time for a few minutes more, John.
Okay,
we will finish, yes.
Okay, so I'll keep going for another couple of minutes then. So I wanted to just remind you about nephronophthisis, that phenotypically it presents with this progressive renal failure, but this polydipsia, polyuria, growth retardation, and anemia is associated with this. That's why actually the first case, the severe hypertension, is quite unusual for a nephronophthisis case, because as they lose salt and can't concentrate the urine, they don't tend to have this hypertensive phenotype, but I guess anything's possible. The corticomedullary cysts and loss of corticomedullary differentiation is quite commonly seen. That's hypertension I mentioned. And really, the biopsy can suggest nephronophthisis, but it's never diagnostic and gets confused with end-stage kidney disease. And so the genetic testing is the only way you can definitely make the diagnosis, in my opinion. And in 10% of cases, nephronophthisis has all these extra renal manifestations, including blindness and cerebellar vermis changes. It can have Bartter-Biedl type associations. Jeune syndrome, as I mentioned, with TTC21B can occur. And Meckel-Gruber is this perinatal lethal syndrome that can have the kidney disease as part of the manifestations. There's probably 19 genes that cause nephronophthisis, and again, about 100 genes that cause the various syndromes that can go with nephronophthisis. And this number of genes is increasing as various genetic labs find new genes. But actually, NPHP1 in the second family is the most common genetic cause of nephronophthisis, and is probably the one that's most easily overlooked if you don't look for the deletion by using the right assays. And in fact, 25% of cases of nephronophthisis will have that NPHP1 deletion. And there's a lovely paper here looking at a cohort of adults with end-stage kidney disease, and by just looking for this deletion alone, the prevalence was 0.5%, which doesn't sound a lot, but these are people who've reached end stage without an accurate diagnosis, and so this single genetic change accounts for a large proportion of these patients. And this paper was published from Nini's group. So this is the Omen data on nephronophthisis, listing all the genetic causes. I say NPHP1 is that, the lion's share of them. The rest account for 2 or 3%, if that. And we've actually got, a good proportion of patients, maybe 60%, that remain unsolved, probably because we've not looked at the available genetic data properly. So this shows the unsolved fraction. This is probably a few years old now, but it shows that, you know, each new gene that's found only takes off a few low percentage of the missing part of the pie. And so there's still plenty of work for us to diagnose nephronophthisis and related conditions. So an algorithm to look for it might be just a simple PCR-based approach to look for the NPHP1 deletions in the first place. And then panels, I think, probably would go rapidly out of date for the nephronophthisis. And I think whole exome and whole genome remains probably the fastest way to diagnose nephronophthisis. But any algorithm that you come up with may be regional or country specific. An example of this was my PhD student who worked in Oman found that a single sort of founder allele in PKHD1 that causes ARPKD was occurring in something like 80% of all patients with cystic kidney disease. So rather than do a panel or exome or a genome on any patient, a simple PCR looking for this founder allele in affected patients proved to be a very cost-effective and efficient way of doing a diagnosis and then widening the scope following that. Okay, and this paper very recently published just points to the advantages of whole genome sequencing, that you can find deep intronic changes that actually lead to aberrant splicing or things that wouldn't be seen on whole exome or Sanger sequencing of exons. And so as the more we do whole genome sequencing, the more we'll find these deep intronic changes. CEP290 has another deep intronic change, but the more we discover these intronic variants that are actually leading to pathogenicity, will increase our diagnostic ability for this condition and others. And so this algorithm was in Emily Grootman's paper. And so again, she relies heavily on the clinical phenotype to direct what approach you'd go. And so, you know, once you've defined that phenotype, it would take you through either a single gene or a limited panel approach, or to this microarray approach if there's multi-system disorders expected, or if the panel's negative or you're suspecting heterogeneous condition like nephronophthisis, you might go for a whole exome or whole genome approach. And what I haven't talked about today and remains a hot topic is what to do with those variants, particularly the VUSs. And so people are still wrangling and struggling to do what would we do with these variants that have never been seen before, in silico, are predicted to be perhaps pathogenic, but remain to be convinced in an in vitro assay. And so to find an in vitro assay for every variant that we're worried about would take many years. So we need to be smarter in terms of looking at transcriptomic information related to the genomic information to allude to pathogenicity. And so that is the work for the next few years, I think. And ultimately, once we have a genetic diagnosis, what do we tell the family? What does it help us do? This is, again, another topic for another day. So I think I've come to the end of that. So thanks for listening, and we'll have hopefully an open discussion time.
Thank you, John, very much for this very comprehensive and excellent overview of genetic testing in inherited kidney disease, nicely illustrated with the very interesting cases. So thank you very much for that. So in our the next 20 minutes or so, we have time for questions and answers. And I'd actually like to start with a few questions that were already asked before this webinar. And the first one is, and I think I start with you, John, the question is, when should we conduct genetic screening in adults with glomerulonephritis and/or chronic kidney disease? John, please.
Yeah, so I'm probably not the best qualified on this panel to answer this question, but I would start by saying, you know, you have to be convinced there is a inherited disease. So, you know, we know there's many types of glomerulonephritis, but, you know, the family pedigree and the information regarding the likelihood of this being either a dominant or recessive cause of glomerulonephritis needs to be taken into account. And I think in terms of glomerular disease, the renal biopsy is still very informative in terms of thinking about what genes you're interested in. I guess FSGS is a good example in that, the most common molecular genetic cause of FSGS now is the Alport syndrome genes, COL4A5. And so we're still learning about what different genes cause which different phenotypes. So I think for glomerular diseases, which I'm not an expert on, that genetic testing becomes very powerful to provide a diagnosis, to avoid lots of immunosuppression where it's not appropriate, to allow transplant decisions to be made. But I'm happy for others to chip in now.
Yes, and maybe just to comment on what you said about the renal biopsy. As you know, it's always a little bit of a discussion what to do first: genetic diagnosis, renal biopsy, maybe prevent even a renal biopsy. So what is your view on this? Or is that also very, dependent on the case?
Well, I think certainly in adult practice, I think we're doing less and less renal biopsies, is my experience, because, if you're smart, you can make a molecular genetic diagnosis before the needle comes out. And this is, I think there's maybe a question about Alport syndrome, but I think if you make a molecular diagnosis in an Alport family, say it's a dominant form of Alport, then it prevents many over investigations and invasive investigations such as the renal biopsy in many other patients, because you can screen for that disease causing variant at the genome level.
Thank you very much. So a next question, maybe Aude, I could ask this to you. So the question is actually, what factors would lead to range genetic testing in unexplained chronic kidney disease in a 40-year-old with no family history? So you have a 40-year-old with chronic kidney disease, there's no family history. What should we do? Should we do genetic testing anyway? And if so, what tests should you advise to do?
Yeah, of course, the familial aspect is very important, because, of course, if there is a familial aspect, I think there is no discussion, and genetic testing should be done. Then now, more and more in adults, we propose a genetic testing in adults with chronic kidney disease, I would say severe chronic kidney disease, before 45 years of age, I would suggest, without any explanation. Of course, first, I think it's still important to be a clinician, so to go to the phenotype and to have a good phenotyping of the patients, searching for any extrarenal complications or extrarenal manifestation. So I think it's still very important to have a good ultrasound or imagery of the kidney to search for cysts and everything, of course. But then if it's still unexplained, and if renal biopsy cannot be performed because the patient has a very severe renal disease, we would perform genetic testing. Then the type of genetic testing depends probably on what is available in the different countries. In France now, we propose to have a very wide panel with we call genome, with all the renal genes that can be found in clinical situation.
Yes.
But then probably in some situation now we can also propose a wider whole exome sequencing for this kind of situation, because then the patient will be probably transplanted, and then it has some clinical impact for the patient to have the right diagnosis, especially if there is a liver live donor for the patient from the family. So the last complication for this patient is also, if we go to the whole exome sequencing, is to have also maybe the parents, because it can help the interpretation of the results. And of course, for the adults, it's always difficult to have a trio, more difficult than for children.
Yes. So thank you very much. So for this. Just maybe to add to this, I know that Dr. van Eerde is also one of the participants, and she, in her group, recently published an article in NDT, also showing that in patients with, onset of CKD, end-stage renal disease before the age of 50, even without, any other criterion to select patients, that the diagnostic yield was actually still very high in those patients. So that really actually emphasizes again the importance of doing these tests in those patients, even if there is not a family history. So thanks very much. While we go to the third question that was also pre-asked, maybe I can challenge you to come up with other questions in the Q&A, because it's still very empty there. And then I'll go to Dr. Plaisier and ask her the third question. And the question is, should we test kindreds of autosomal dominant Alport disease with microhematuria?
I think, yes, it's a very important question, an actual question regarding the expanded data showing that mutation in COL4 genes are involved in more and more renal diseases that are not the so-called Alport syndrome, which is classically associated with extrarenal manifestation. Mm-hmm. And in the more recent year, we see pathogenic variants associated with glomerular disease that were not diagnosed as hematuric glomerulopathy. So I think it's a very important step first to for glomerular disease like unexplained and cortico-resistant nephrotic syndrome, or a chronic glomerulopathy with an undetermined clear classification at the renal biopsy. It's very important first to classify the disease for the patient. Second, to search for relatives with polysymptomatic or undiagnosed glomerular disease, because we now know that some of these patients with heterozygous variant in COL4 genes are associated with progressive renal disease, even in the late after 50 years. But it's very important now to give all the recommendation to prevent progression of the glomerular disease. So I think is now we have to ask for testing a patient for this variants in collagen 4 genes in patients with unexplained glomerular disease, even without hematuria, because we now know that some patient doesn't have hematuria, even if the glomerular basement membrane seems to be normal or only thickened at the electron microscopy. And now we know that in some patients classified as having IgA nephropathy, with or without familial history, show pathogenic variants in the free collagen 4 genes. It has been repeatedly published in the last year. And we also know that the histopathologic diagnosis of IgA nephropathy is sometimes based only on the presence of IgA by immunofluorescence and without mesangial proliferation of hypertrophy. So we have also to think about this diagnosis in patients with IgA nephropathy, and mostly if there is another patient in the family, another case with so classified IgA nephropathy.
So fully agree with you, and not yet mentioned, but of course, if you sort of reclassify these diseases, it's not only that it's maybe important in terms of of therapy, but or potential therapy, but also in terms of, let's say, genetic counseling and reproductive options.
Yeah.
So, you know, if you're dealing with an Alport syndrome, it's quite different from IgA nephropathy in terms of genetic counseling. So we shouldn't forget the importance of having the right genetic diagnosis in this case. So John, I have both a comment and a question from Dr. van Eerde. First of all, she thanks you for the nice overview. And she said, well, what I just want to mention is that if you have whole gene NPHP1 deletions, these can be detected in next generation sequencing reads if you really look for them. And I think that's right. Huh? And the, you know, the better the NGS platforms become, the more easy it becomes to take, you know, to pick this up from these NGS tests. So hopefully in future, you know, we do not need to do arrays or whatever for finding these whole gene deletions. You agree with that, I guess? Yes.
I would agree with that, but I think historically the absence of any variants in the gene, you think, well, that gene must be normal, but the absence of the complete absence of any polymorphisms in the gene, you suspect, well, where is the gene? It must be deleted. So I think, yeah, our interpretation of this data is getting better now.
Yes. Okay. So and then a question she has. So if a nephrologist identifies a variant in a patient with genetic testing, what are the arguments for you as a nephrologist to refer to a clinical geneticist, and what are the arguments against?
Yeah.
So when do you not refer?
Yeah.
Coming from a clinical geneticist, this question, huh?
Yeah, this is a really hot topic, and I think, you know, we've got to get better at working with our clinical geneticists. As you alluded to before, you know, CKD, chronic kidney disease, you know, might be a genetic disease a lot of the time. And so, you know, this compartmentalization of I'm a nephrologist, but I don't deal with the gene stuff, that's someone else's problem, needs to be resolved. And I think every nephrologist needs to be aware that, you know, at least maybe half of what they see could be genetic disease. But again, we need the clinical geneticists to help us understand and develop expertise in understanding the variants. So I think the role is for MDTs is essential, really, because, you know, whenever I have an MDT, I'm learning new stuff from the clinical genetic side, and I think they learn stuff from me from the nephrology side. So we've got stuff to teach each other about the management of these patients. Yeah. I fully agree with that. So in a multidisciplinary setting, working for these patients is the best way. So then there is a question or comment, maybe? No, a question from Dr. Rodriguez. She says, I follow a family with microhematuria, atypical cystic disease, hearing impairment, and end-stage kidney disease with autosomal dominant inheritance, at least 3 generations, all adults. They are negative for the PKD genes and the Alport panel. And she asks, and maybe this is a good question for Aude again, is exome testing the next step? So she has done the panels for Alport and PKD genes. Would you then go for exome?
First, yes, it could be Alport patients, Alport family. So
yeah, but she did the panel.
Yeah, the panel is negative. But
yes, she says
another option should maybe to have some skin biopsy for fibroblast and look for RNA. But if it's available. Because to search for intronic mutations and other mutations that can be found by panels. But the other options first, maybe whole exome sequencing to search for any other kystic disease.
Yes, yeah, that could be an idea. And it depends a bit on the family, but you could still think about the old-fashioned linkage analysis first to see whether you can see whether there's some linkage to a certain genomic region that would bring you to give you an idea about what to think about. But or combine exome sequencing with linkage analysis. That could be a next step, actually, in my view. Yeah. Okay, she adds, one patient had a thin basement biopsy. Would that help for you?
Yeah, go for the RNA.
Yeah. And Albertine van Eerde adds, hereditary deafness could also be screened. So a lot of suggestions to Dr. Rodriguez, and we hope that this helps her. Yeah. So there are no further questions from the Q&A yet, but there's still some time left. And actually, I've maybe another question for John. So John, you are in the lucky situation that you are in England with the NHS. And for instance, in the Netherlands, we are in a very happy situation that genetic testing is just in the basic health insurance. So everybody can get a test, that will not have to pay for it. However, in many countries, this situation is very different. And for instance, in the United States, all genetic testing is done by commercial labs. They have to pay a lot of that for having this done. And of course, let's say in less developed countries, the access to genetic testing is really, really bad. And there is even no access. Let's do not talk about costs anyway, but there is no access to genetic testing. So what in your view can we do to solve that situation? That every patient in the world has the same access to genetic testing. Or is that just not possible at all? I mean, what could we do coming from this very lucky countries?
Yeah, it's a important question. I'm not sure I have the answer. I think there is this blurring, as you know, between what a clinical genetic test is and what a research genetic test is. And I think many countries do rely on interested research laboratories to kind of shortcut their way to a gene test. And I think that is acceptable up to a point. But I think, in terms of equity, eventually the cost of exome genomes is going to be much lower, and so will allow rich countries to subsidize genetic investigations of poorer countries. But who coordinates that and
exactly
allows that to happen, I'm not sure. But I would like to see within the next 10 years that there were ways of that was happening, and that everyone with suspicion of an inherited disease got access to the right testing.
Yeah, I fully agree. And I think this is also something that we could discuss with the International Society of Nephrology, who is very much involved in, trying to get everything also to less developed countries. So, but it's something it keeps my mind occupied all the time, because we are so lucky that we have this situation and others don't. Well, it's 2 minutes. Sorry.
Yes, another thing.
Dr. Plaisier, yes.
Yes, we are on the story, because probably in the near future we will learn from what we are now going to make a genotype phenotype correlation, and go back to the clinic, and to not usually go the ultimate goal is not usually to go to the whole genome sequencing, but to learn about what we are discovering now, to go back to the clinic and develop algorithm based on a very good phenotyping, and to go in the right way on the group of gene, and not going to the whole genome. I think it's not the ultimate goal. We have to learn about what we are see now with the genetic data that are accumulating with all our work. I think it will be important to go back to the clinic and to go to phenotype, to good phenotyping and clinical biomarker or histological biomarker that will be very important to simplify the diagnosis in the future.
Thank you very much. So maybe just one last question from Dr. Hashemi. He says, he or she asks, which time will we do genetic testing in FSGS? I'm not really sure what he means, but the question, I think the question is, do we need to do genetic testing in FSGS? John, what is your opinion?
I think before the patient's exposed to, chemotherapy and, risks of recurrent disease and transplantation and everything that ensues, that I think genetic testing, should be done as soon as it's suspected, is my view.
Yes, thank you. Thank you very much. So thank you again, John, for this very nice e-seminar, and the panelists for being involved in the questions and answers. I think we had a lively discussion. So I'd like to thank the participants very much for attending this e-seminar, and I'd like to ask you to stay connected for 1 more minute to complete a short feedback survey that should not take you more than 1 minute of your time. And with that, you provide us with very useful information so that we can help, that we can improve the quality of our e-seminars. So if you would do this, we would be very happy. So thanks again for your participation, and thanks everybody from the panel and the speaker again for this great seminar. Thank you very much, and bye-bye.